So welcome to the ARO seminar series centered on the topic of auditory nerve. Today we are delighted to have Dr. Christine Köppl as our speaker. I'm Ken Henry and I'll be your moderator for today's seminar. So during the presentation, if you have any questions, please use the chat function directed to me as the moderator to avoid interrupting the presentation. Following the presentation, in addition to using the chat function, also feel free to raise your hand using the Zoom function to directly ask questions. Any questions that we're not able to get to during the session, which will conclude at 1 o'clock, Christine can answer those by email afterwards. Closed captioning for the meeting is available for your use. And the session is being recorded and will be made available to be viewed later on. If you have any technical difficulties using the Zoom interface, you can email ARO at headquarters@aro.org for assistance. Okay, so Dr. Köppl has a background in zoology and neuroscience. Her long-standing interests are in the evolution of the auditory system and have included comparative work in mammals, birds, and reptiles from the level of the cochlea to the midbrain. She has published 115 peer-reviewed papers, including 31 review articles. Christine received her biology diploma, equivalent to a master's, in 1985 from the Technical University of Munich. She did her PhD work also at Technical University of Munich with Jeff Manley on inner ear anatomy and physiology in lizards. She remained based in Munich for several years with research trips to Australia to work on lizards and to the US working on barn owl sound localization. After a prestigious Heisenberg Fellowship by the Deutsche Forschungsgemeinschaft, which is the major grant funding body in Germany, Christine moved to a faculty position in the Department of Physiology at the University of Sydney in Australia in 2006. In 2009, she returned to Germany as a full professor at the Karl von Oschitsky Universität in Oldenburg. There she was part of the larger cluster of excellence, Hearing for All, and her research there focused on age-related hearing loss and cochlear aging, primarily using the Mongolian gerbil as an animal model. Most recently, she's added exciting new data from aging birds, which in contrast to mammals, are able to regenerate their sensory hair cells. Christina has recently retired and is working to wrap up publication of her latest projects. So without any further delay, it's my pleasure to welcome Dr. Köppl, who will be presenting her work titled Beyond Mammals, Auditory Nerve Responses Across Different Species.
Thank you very much, Ken, for this kind introduction. I'm happy you agreed to be the moderator today. I should say I don't see the Zoom screen, so if anything is going wrong or you can't follow or the slides don't project, please let me know by speaking up. Okay, so it's a great honor to be part of this great lineup of speakers on auditory nerve. And I'd like to start by thanking the mentors who introduced me to this work and to the field of non-mammalian auditory physiology. There's first and foremost, first and foremost, of course, Jeff Manley, who's taught me everything I know about recording from the auditory nerve. As a master project, he gave me the task to record from European lizards whose auditory nerve has about 200 nerve fibers, and I suffered through finding at least a few of them. Later, he put me onto recording from the auditory nerve of the barn owl. And it was in Mark Konishi's lab that I laid first eyes on the cochlea of the barn owl. I also met Catherine Carr in this lab, and she's become a very good friend and a highly valued collaborator to this day. And we've done a lot of exciting barn owl work together. So I will focus today on responses from the auditory nerve, so on the physiology of auditory nerve fibers, and this is what I aim to cover. I will start, at the risk of being a bit trivial, I will start with pointing out some really basic shared properties of auditory nerve responses that are apply across all vertebrates. This is to take out maybe the exotic of it and put everyone on the same page about what is actually shared. They're not that different. They're not totally radically different from mammalian responses. But I will keep this brief and will spend most of my time talking about birds, introducing first a few backgrounds that we need to know to interpret, to know what we record from when I talk about auditory nerve fibers, and then summarize the current knowledge about what I think are very interesting differences in auditory nerve responses between birds and mammals. And I think this summarizes it quite nicely. Are we looking here at the same thing but different? And if we have time, I will conclude with some of the latest work, as Ken mentioned, about aging in the bird auditory nerve. Okay, a lot of shared properties of auditory nerve responses, of course, go back to the input from the ribbon synapse. And I'd like to emphasize that all auditory nerve fibers across all vertebrates get their input from ribbon synapses. I've collected 3 examples here from hair cells from different groups. There is one from a frog here, one from a turtle, and this is an actual confocal image from a bird auditory hair cell. And you can see in this bullfrog example, there's 2 afferents coming up to these hair cells and engulfing it by finger-like projections. Very similar here in the turtle hair cell. There's also the ribbons as little dots here indicated. And you can see there's quite a number of ribbons feeding into each afferent. This is the same in the bird. The gray thing here is the hair cell. The cyan terminals, there's 2 or 3 coming up to these hair cells, and each, as you can see, labeled by the yellow dots, each of these terminals gets input from several ribbons. So all auditory nerve fibers get input from ribbons, but in non-mammals, typically, the relation is not one-to-one, as in the mammalian type ones, but several ribbons, many, and sometimes many ribbons, like here, 53 actually, feed into one afferent. And I will quickly go through this list, illustrating, showing a few examples, mostly from non-birds, to mainly make the point that there are also quite a lot of classic data out there that you can find if you're interested on all major non-mammalian groups of vertebrates. Of course, the first one on this list here, the first characteristic that is shared across all auditory nerve firing is that they all show irregular spontaneous activity. And this, of course, goes back to the synapse. So spontaneous transmitter release from the ribbon synapse drives spontaneous discharge in all auditory nerve fibers. Also, a shared property is that they all increase their spiking discharge once the stimulus is above threshold. Auditory nerve fibers universally increase their spike rate when the sound level increases, and you see here an example of a rate level function. So sound pressure level versus the average discharge rate. In this case, an example from a lizard. The exact shape of these rate level functions might vary, and we will get back to that later when talking about birds. But the principal rise and eventual saturation is a universal characteristic. Also, if the if a constant stimulus is longer than, say, about 10 milliseconds, adaptation will be apparent. And here you see an example from the chicken, in this case, a peristimulus time histogram. So probability of occurrence of spikes over 100 millisecond stimulus, constant tone burst of constant amplitude, and you clearly see the adaptation. So the initial discharge rate is high and adapts then to a quasi-steady state when the stimulus continues at a uniform amplitude for some time. Again, this is a shared property, and this in particular, of course, also goes back to the synapse, and the time constants and general rules are the same as in mammals. Phase locking is also a universal shared property of auditory nerve fibers, at least those that are sensitive to lower frequencies. And this is not so intuitive, phase locking. That's why I put up a small schematic here showing you or illustrating graphically what it means. So if we record from an auditory nerve fiber that connects to a hair cell and record the train of spikes, and then have a closer look as to the temporal relationship of these spikes to the sinusoidal waveform, if it's a simple tone, but one can also do this with more complex stimuli. Then it's not so easily apparent, but you can also extract phase locking behavior using noise, for example. But in the simplest case of a sinusoidal tone, an ongoing sinusoidal tone, if a fiber phase locks, you will realize that the spikes tend to occur at certain phase, at a constant, always the same phase of the stimulus. Now, this is biology. This is not a technical system, so there will be some jitter around this average phase, and this can be then quantified as a phase histogram. So the probability of spiking again, but not as a function of the entire stimulus duration, but only as a function of one cycle, one period of the stimulus. And if phase locking occurs, the spikes tend to cluster around a mean phase. This is an example from the barn owl at a frequency of 5 kilohertz. This is very high already. I've said phase locking is limited to low frequencies. So low in this case means a couple of kilohertz typically. And here you see another example from a lizard. This is not a phase histogram, but again, a peristimulus time histogram. At the high level, you clearly see the phase locking. The stimulus here was not phase constant, so it did not start with the same phase in each trial, but you can clearly see the lineup of spikes in successive trials. And finally, frequency tuning and tonotopic arrangement is also a universal characteristic of the auditory nerve and the hearing organ wherever you look in vertebrates. You have 3 examples here, in case 2 from 2 different lizard species, tuning curves, so frequency as a function of sound pressure level at threshold, and each of these V-shaped curves is the response of one single auditory nerve fiber. You see they're all basically V-shaped, sometimes slightly different shape with a tip and a tail, sometimes very sharp and V-shaped, but frequency tuning is a universal characteristic of all auditory nerve fibers. How the tuning is achieved might differ, but that's not the subject of today's talk. That's more about cochlear mechanics and less about the auditory nerve. So these 4 basic characteristics we find throughout all vertebrates, and I've put a small, what I call a starter reading list for you together. You can browse this at your leisure later if you're so interested. These are reviews, and I've selected them specifically because each of them says something about the auditory nerve. So this is not just about comparative hearing in general, but I've tried to find papers that really discuss auditory nerve. Because I won't talk much about non-birds today, I thought it was important to maybe give you a start if you're so interested. Okay, so move on to birds. But before we do so, I'd like to emphasize one point, one main difference that we need to be aware of, that we should be aware of between mammals and non-mammals, and that is the frequency range of hearing. So mammals in general hear much higher frequencies than other vertebrates, and what I use here to illustrate are some very average audiograms from a great compilation by Dick Fay in the 1980s. So the different colored curves here show average behavioral audiograms for these different vertebrate groups. So from fish in gray, amphibia in green, birds and other non-avian reptiles, they're all lumped in this orange curve, and mammals in this light blue. And you can easily see there's a pretty significant difference here. So all the non-mammals are confined to below 5, 6, 8, 9 kilohertz sensitive hearing. Anything above is only found in mammals. But, and there's a caveat I'd like to make, there is really no such thing as the average mammal. And I'd like to highlight here that humans are actually not so typical or not so characteristic for an average mammal. So the black audiogram here is the average human audiogram, and in blue are a couple of other mammals. Most of them go much higher, as you see, and there is the mouse, which is a high frequency specialist. And in orange again, the same audiogram, same average audiogram for birds here. The point I'd like to make here is that for even in a biomedical context, if you're primarily interested in human hearing and questions of disease and hearing loss, for many questions, non-mammals may actually be better animal models than the mouse, for example, because the hearing range is much more similar to a human. And birds in particular are attractive because many of them can be trained and behaviorally tested fairly readily. And I'm sure that's one reason why our moderator, Ken Henry, today works on budgerigars. And of course, the press picked up on that long ago. So just for fun, here are some press clippings. Some of them go a long time back. You see very useful renderings of myself and Jeff Manley here together with Graham Yates in Western Australia, and some press echo that was more recent when we published our work on the ageless ears of birds. Okay, so let's get to birds. This is just to remind you that only the mammalian cochlea is coiled. That's why it's called a cochlea, the snail. So here is the inner ear of a typical mammal, and here contrasted to the inner ear of a typical bird, a pigeon in this case. And in red, the auditory epithelium is highlighted. So of course we have cochlea here in a typical mammal. In a bird, this looks much more simple. It is more straight, more like a banana shape or a boomerang. That's what it's sometimes likened to. And the term we use for it, or the term I will use for and prefer, is actually basilar papilla. It's mostly historic that terms are used. The terms that we use for mammals and non-mammals are different, but cochlea literally means snail, and so strictly speaking, one shouldn't use the term cochlea for any non-mammal. But these are homologous structures, so they have the same origin, they go back to the same roots. So I will talk about basilar papilla when we talk about birds. The basilar papilla, the hearing organ of birds, is typically a couple of millimeters long. That's a typical number you can remember. And this is a classic figure from a paper by Schwarzkopf and Winter in the 1960s already. They did a large comparative study looking at different kinds of birds. And you see the outlines of the basilar papilla here for a number of different birds. Chicken up here, pigeon down here, a raptor or a bird of prey, a couple of songbirds here. And this is, I believe, a parrot. So they're all a couple of millimeters. That's a typical size of bird basilar papilla. Schwarzkopf already also looked at the barn owl and pointed out its enormous length. And for fun, I'll throw in here the barn owl because I will also show you some data from this bird, although I will not dwell much on the specializations of the barn owl cochlea today, but I'd like to mention at least it is special, it is especially long, and it's a fascinating system. So this is the outline of the barn owl cochlea, and it's actually quite three-dimensional, although it's not coiled. It has a complex three-dimensional shape, as you can maybe appreciate in the scanning EM photograph down here in purple. The basilar papilla is highlighted, and it actually goes out of view and curves in an almost half turn down out of sight. And this top here is a cool CT scan-based reconstruction that I got from Stig Walsh from the National Museum in Scotland a while ago. You can see that the two cochlea actually meet at the midline here. The apical ends meet in the middle of the skull. But so coming back, what is typical for birds, and this also applies to the barn owl. The types of hair cells differentiate in a kind of similar way to mammals, but not quite the same. So this is the first illustration, or a good illustration, I think, of what I mean by same thing but different. We have, again, the outline of a EBU basilar papilla here in this case. So just the few millimeter long outline, the apex up here, which has low frequency responses, and the tonotopic gradient going all the way to the base, to the narrower base, which represents high frequencies. And here you see two representative cross sections. So these are actual histological cross sections. You can just make out the hair cells here as these darker structures as we go across the tectorial membrane up here and the nerve fibers coming in from this side. So this is a representative apical cross-section, this much more basic. And you see at first glance, this is quite different in some respects from the mammalian cochlea, which I assume you're sort of familiar with. So this epithelium is wider in the apex, and it also has a lot more hair cells across at the apex and at the base. So there's no uniform hair cell number at any one place across. And to the right here, you have schematic drawings of typical hair cells as we go from the inner neural side to the outer abneural side. We call them tall hair cells in the apex, and then this gradually transitions to short hair cells as you go towards the base and across the papilla. And the really distinguishing feature between tall and short hair cells is their innervation. So in that sense, they're quite similar to mammalian inner and outer hair cells. Tall hair cells get afferent innervation. These are drawn in blue here. Not as many as in a typical inner hair cell, so only about 2, 3, 4, 5 on one tall hair cell. And they also get efferent innervation, directly somatic onto the hair cell. Short hair cells get only efferent innervation. And that's what gave them their name. They're typically shorter than they are wide. And I won't dwell on the definitions of tall and short hair cells. We can talk about that if you're interested in the question session. My definition of short hair cells follows the one suggested by Fischer, who did a lot of transmission EM work and who made these nice reconstructions and schematic drawings. So a short hair cell is one that has no afferent connection. And the important thing to remember, this is different to mammals. There is a continuum between tall and short hair cells along 2 axes. So there's a transition from tall to short as we go across, and also as we go from the apex to the base. Short hair cells will increase in proportion towards the base of the bird basilar papilla. So coming back or reminding you about the recordings I'm going to show you, all the auditory nerve recordings we're going to talk about are from these blue terminals here. And just for fun, here is a real microscopic and immunohistochemical stained cross-section in two parts. These are actually not even all hair cells. This is from the apex of the barn owl. And so here, this is not special in any way. This would look the same or very similar in a chicken or a pigeon. You have tall hair cells on the one side, and these slowly transition to shorter hair cells on the outer side. And you can also see the transition to these large terminals, these yellowish labeled efferent terminals here, increasingly on the short hair cells. The barn owl is special in, and this is the last anatomical slide I'm going to show you. The barn owl is special in that in the so-called, what we call the foveal region, that carries a large overrepresentation of an important high-frequency band for the barn owl. The hair cells receive a lot more afferents than is usual in a bird. So this is a similar schematic as I've just shown you for the emu, but for the barn owl, the much longer basilar papilla with the apical end here, the basal end down here, and again schematic illustrations of hair cells as we go across. And you see that in this mid-basal region that we call the foveal region, there's many more of these blue afferent terminals on each tall hair cell than we see in a typical bird. Okay, let's go to physiology. The first point I want to make about auditory nerve physiology in birds is that tuning, we've already mentioned, tuning is sharp, tuning is nice, and the tuning curves are rather simple V-shaped. There is no tail. This is a distinct difference to mammals. So this is a collection of tuning curves from the barn owl, but you could take this from any other bird, except that these highest frequency ones would be missing. So the barn owl extends to somewhat higher frequencies than most other birds, but in any other respect, these tuning curves are entirely typical, nothing special. They're not especially sharp. They're very sharp, but they're not especially sharp for a bird. Oops, apologize. Some delay just involved. Okay. Spontaneous rates. I've mentioned spontaneous activity is characteristic for all auditory nerve fibers in birds. All auditory nerve fibers is what in a mammal we would call high spontaneous rate. And the distribution of spontaneous rates is monomodal. So what you see here is examples from 4 different bird species: chicken, pigeon, starling, songbird, and the barn owl. The range here is up to 200 spikes per second in bins. And you can see this is near normal distribution, a bit species specific perhaps, but there is no distinct category or no distinct peak at very low rates, as Charlie Lieberman showed you beautifully last week for a typical mammal. What is interesting and maybe a bit surprising at first is that the threshold distribution is typically very large. So what you see here are large data populations, again from chicken, pigeon, and starling, where the characteristic frequency, so the best response frequency, and where threshold at CF is plotted as a function of characteristic frequency. And you will appreciate the large variation. So the axis here covers 90 dB SPL, and in each case, the data population varies from near zero up to 80 or 90 dB SPL. This got people initially worried, and every one of the classic early studies that recorded from birds went to some lengths to show that this was not due to the animals being in poor condition. So this is quite well accepted, and the evidence is good for it. This is normal. The only perhaps interesting exception is the barn owl, where the threshold range is a lot lower. That's an interesting oddity for now that I won't dwell on, and I can't really explain yet. I'll take it with a bit of a grain of salt because the low frequency sample here is very limited. So there may be, one could speculate that there may be a larger spread at low frequencies, but definitely at the high frequency in this foveal region, the spread is much smaller than in other birds. Now, despite this large variation, there is no clear correlation of sensitivity with spontaneous rate or the rate level function, which I'm going to show you in the next slide. And I'm sure you are aware that such correlations are very typical for mammalian type 1 auditory nerve fibers. So there is no such clear relationship in birds. And again, examples, large data populations from 4 different birds: chicken, pigeon, starling, and barn owl. For the chicken and pigeon, there was a slight correlation, a weak correlation of spontaneous rate and sensitivity. But look at the variance overall. So this is nothing like what you would see in a mammal, and there was definitely no correlation at all statistically in the starling and in the barn owl. Rate level functions look at first sight very similar to what you would see in mammalian auditory nerve. We find the same 3 types that have been classified or defined in mammals, and you see examples here from the chicken. So the classic saturating nerve fiber with a steep increase, break, and then constant rate that doesn't increase anymore as you increase the sound level, and the so-called sloping saturating fibers that still increases, albeit with a shallower slope above this break point, and then the straight one, which does not seem to saturate within the sound pressure levels that are typically tested. Examples from the pigeon showing the same 3 types, and also in the barn owl, except here the straight type. I'm still not sure if it really occurs or if it's just extremely rare. I've seen it only once in this larger sample that we reported in this paper. So I have left this off. This does not seem to be a typical type in the barn owl, but saturating and sloping saturating definitely. So at first glance, this looks like there might be similar correlations, but they are not. If you plot again threshold sensitivity for large populations of auditory nerve fibers as a function of characteristic frequency, and now differentiate between those with the 3 different types of rate level functions, there is absolutely no relationship. You might be hard pressed to distinguish these symbols here, but don't even try. There's nothing to see. There's complete overlap between the 3 types of rate level functions, same for the pigeon and same for the barn owl. In the pigeon, there was a slight trend, perhaps for the straight rate level functions to be a bit less sensitive on average, but the spread is large, even for those. So there's some down here as well. And that remains a bit unclear, but definitely for saturating and sloping saturating, there is no relation to threshold. So by now you might be thinking, well, this is all very interesting, but if we can't explain this, what about it? But I think what helps us solve the puzzle of why birds are so different, or why these correlations that we see in the type 1 fiber populations in mammals are not seen, was at least partly answered by two classic studies in pigeon and starling, which I summarize here. Now, these two studies did the hard work of recording from single units, followed by dye injections, and then follow these histologically to their point of innervation in the basilar papilla. And what you see here is an example, three examples from the pigeon study by Smolders, where the three fibers with comparable CF, these are all around 700 Hertz characteristic frequency, but different sensitivity, were then shown to innervate hair cells at systematically different positions across the width of the papilla. And that's what you see schematically drawn here. So these shapes here are hair cells as we go across from the inner side to the outer side. So here would be the short hair cell zone, and to the left, the neural side where the nerve fibers come in, where the tall hair cells sit. And you see that the most sensitive nerve fiber connected with a bit of a distance here, but very close to the inner edge. The lesser sensitive fiber somewhere in the middle, and the least sensitive fiber out here. And this turned out to be typical when they looked at the entire data population, all fibers that they had labeled, and they, because this encompassed a large CF range, they normalized the threshold here as a relative threshold to the expected average value, and drew a running average through these data points. So here we have sensitivity, a metric for sensitivity, as a function of where this fiber innervated across the papilla. So here was the inner edge or the neural edge, and out here is the outer edge where the short hair cells sit. So one interesting point here is very few fibers did connect out here where the short hair cells are, which is consistent with what Fisher found anatomically, that the further you go out here and the shorter the hair cells become, the fewer afferent fibers you have. But this running average here was very interesting. What they concluded from this data was that there is a strip of hair cells, which is some distance from the inner edge, which is the most sensitive ones, and their associated fibers. Very similar data from the starling. In this case, sorry, I'm having a slight delay here with the pointer, which is a bit annoying. Similar plot for the starling, except here the threshold variation across the width was clearly just rising. So it looked as if the most sensitive hair cells and their associated fibers were sitting right at the inner edge where the nerve fibers come in. And the further out the hair cell was situated towards the short hair cells, the higher the thresholds were. The threshold change was quite clear in both species, albeit a bit different. The correlation with Q10 was much less clear. So this is a bit of an open question. In pigeon, it approximately covaried with the threshold, but in the starling, there was no correlation at all, and spontaneous rate did not correlate with any of this in both species, again confirming that the classic subgroups that we see in mammals are not present in the bird. Instead, and this is my summary of this bird auditory nerve physiology, and reinforced by exciting new data, I think, about the molecular profiles of hair cell types in the avian basilar papilla. This is a schematic I've taken from a paper from Stefan Heller's lab, who's done exciting new work on the molecular characterization of hair cells, so not the afferents, but the hair cells. And they have evidence for 3 to 4 distinct hair cell molecular identities as you go across the papilla. So this tallies with what we see morphologically, going from tall to short hair cells, and they have evidence that the tall hair cell, they presented evidence that the tall hair cells subdivide even into a most neurally lying group, which is molecularly distinct from the tall hair cells that are a bit further out, and then again from the short hair cells. And that, I think, ties in very nicely with these classic physiological data, which show that the sensitivity of the hair cells and the associated nerve fibers changes as we go across the papilla. So what this looks like, in summary, is that we have a broad diversity of sensitivity here across the papilla, going from tall hair cells towards short hair cells. How exactly this gradient looks like may be species specific, so that's an open question. I've shown you that for the pigeon it looked more like this, where the most sensitive fibers were a bit removed from the neural edge. In the starling it looked as if the most sensitive fibers connected right at the neural edge. Maybe there's also an apical basal difference in this, so that's still a bit unclear, but it's definitely a system that is set up a little bit different, but in a way also similar to the mammalian cochlea, where the sensitivity is distributed, the sensitivity gradient is distributed among several hair cells instead of all concentrated on one hair cell with different fiber types connecting to it. And then just looking at the time. Okay, I think I have a few more minutes to spend to tell you about some exciting new data on aging. So these were all very important, I think, data, but very classic data. This is all published, what I've told you so far. And I'd like to, for the last few minutes, I'd like to spend on some newer data where we've been looking at aging. And this has a background, of course, in the well-known regenerative capacity of birds. It is quite well known that after damage, birds can regenerate hair cells and can also regenerate their physiological responses in the auditory nerve to an amazing degree. So when it was shown that birds show no age-related hearing loss, and these are two very nice studies also published by the lab of Georg Klump at my university, both in starling and in barn owl. Old birds, quite old birds, show no hearing loss when you test them behaviorally.
We can't see it.
So I was wondering, though, more recently, after having done a lot of gerbil work, how much of this was actually really due to the cochlea. I mean, the assumption is a natural one that due to the regenerative capacity, birds do not suffer age-related hearing loss, and that this has its basis in the cochlea. But is this really true? A behavioral audiogram cannot really trace it unambiguously to the cochlea. So we embarked on a quite large study in the barn owl, looking at a lot of different things, and I will just summarize this in 2 slides because the message is really quite simple. We have found nothing so far. So even at the cochlear level, we have no evidence for any age-related cochlear dysfunction. We've looked first at evoked potential, so CAP thresholds. That's the classic CAP or ABR. And you see the average thresholds here for a group of young adults and a group of aged owls measured in the same setup, interleaved. And if anything, this is not statistically significant, but if anything, the old owls were actually more sensitive than the young ones. We've looked at CAP amplitudes as a metric of synaptopathy. Again, this will be quite familiar to many of you in mammals. This is now a well-known metric for fiber loss, for synapse loss. But in the owl, I find no evidence for any loss. Again, if anything, the old animals have higher amplitudes than the young animals. We've also started counting synapses. I've shown you an immuno earlier on in the talk, and we're counting ribbons and looking at terminals. But this is ongoing, so the numbers here are still a bit low, but you can easily see that at 3 different cochlear locations that we've evaluated so far, there is no statistical difference between the old animals and the young, confirming the physiological data, the CAP amplitudes. We've looked at endocochlear potential, and the tissue that generates it, which is the tegmentum vasculosum in birds. The endocochlear potential is not reduced in old owls compared to young, and the preliminary data on the blood vessel area that is in a standardized area of tegmentum vasculosum also shows no difference between young and old. And at the single unit level, the thresholds, single unit thresholds confirm what we see at the level of the CAP. So again, a data population here, no difference between the young adult and the aged owls. Spontaneous rates are the same. They do vary with CF, and that is already well known. That's published, but there's no difference, no change with age. And also temporal metrics, something which the barn owl is quite famous for, phase locking up to about 8-9 kilohertz. The vector strength as a function of base frequency is shown here. And again, no difference between adults, young adults, and aged owls. And this is another metric for coding temporal fine structure, which also showed no difference. So I'm quite excited about this. This is all unpublished and we're ongoing in our analysis, but I will conclude with that and hope that we still have time for some questions.
Okay, so any questions, please put them in the chat, or you can raise your hand as well, and we can try to do questions that way.
Hi, Peter.
Hi, Christine. I have a question for you. Wonderful talk, Christine. Thank you so much. I was interested in if you have discovered what feature in the mammal is responsible for the tail of the tuning curve, and which is not doesn't exist in the bird, you said.
No, I haven't. So, I mean, in the mammal, it's general knowledge, or I guess it's assumed knowledge that this is due to the traveling wave.
Right.
The way it travels, the dispersion of the traveling wave along the cochlea, and this is still, I would say, this is still an open question in birds. There have been some really good, careful measurements at the level of the basilar membrane, which have shown crude traveling wave in birds, which is linear. So this looks quite boring and disappointing. I would say it probably is the wrong place to look. This is also the wrong place to look actually in mammals, but you're just lucky, I think, in mammalian physiology that the movement of the basilar membrane reflects what is happening at the levels of the hair cells as well. I mean, transduction doesn't happen at the basilar membrane, transduction happens at the hair bundles. And in the mammalian cochlea, the system moves as a unit, and you can measure the sensitive nonlinear motion quite well at the level of the basilar membrane. In birds, you obviously can't. And I don't think this means that there is no nonlinear sensitive movement. There's good evidence for active amplification also in birds. The mechanism is unknown. But whether there is a traveling wave, a dispersing traveling wave, as in mammals, I would say is still unknown. We should measure at the tectorium membrane level, and this has just not been technically possible. No one has tried. It hasn't been done. That's where we might find the real micromechanical motion that matters, I think. This doesn't explain your question about tails, But what I'm saying, I guess, is that the micromechanical motion is probably quite different in the bird cochlea. And we really have no good idea currently how it moves and how the stimulus reaches the hair bundles.
Thank you. Very good.
Great. It looks like we have a question from AOI Ann Hunsaker. Would you like to ask your question? I see the hand up in the chat as well.
Oh,
Okay,
should I ask on the mic? Oh, okay, great. Hi, thank you for your talk. I was curious if you could talk about like the regenerative properties of avian hair cells, like why birds can do it. And I think in my previous classes, we talked about cytoskeleton, actin filament thickness. Yeah, if you could talk about that, that'd be great. Thank you.
I'm not the best person to ask that. I mean, I followed the literature, obviously, and it's still a very active field of research. What we do know is that in the bird, the supporting cells are the source of regeneration. So they can either divide again and produce another supporting cell in the hair cell, or there's also the possibility of direct transdifferentiation, so a supporting cell becoming a hair cell after damage. Why mammalian supporting cells are not able to do this? I cannot really answer conclusively. A lot of research has gone into this. So far, it hasn't really been possible to reactivate cell divisions in the mature organ of Corti, and that seems to be the major obstacle. So I think there's nothing specific about bird cells that enable them to regenerate. It's more the other way around, that in mammals, this has somehow been turned off when the organ is matured, and it's not really clear why, but there's a lot of still significant efforts to try and revive this capability.
Thank
okay,
you.
great. It looks like our next question is from Kazuhiro Hori. Would you like to unmute?
Yeah, can you hear me? Good. Yeah, thank you for a very interesting talk. And I was very excited. Birds don't show no age-related cochlear dysfunction, and I also wonder, have you ever tried acoustic trauma or toxic drug in birds? They also don't show such kind of cochlear dysfunction or not?
Well, they do initially. So there's a lot of classic work on noise damage and also on ototoxic damage. Birds do experience that. Hair cells die. Hair cells die big time, depending on how loud you make it and what the dosage is. The main difference then is that they can regenerate. So the organ is damaged, is severely damaged, but it can repair itself. And it seems that the auditory nerve fibers hang around and wait for the hair cells to regenerate and then reconnect. So the capacity to repair is just much, much greater than in the mammal, but the damage happens.
I see. Another question is, only hair cell can be regenerate, or neuron in birds also can be regenerate. How do you think about that?
This has not been addressed to my knowledge. So the hair cell regeneration is very well known, is very well characterized. We know a lot about the supporting cells, how they regenerate, how they turn into hair cells. The neurons have been much less in focus, but there is some evidence that they obviously detach if the hair cell dies, but that they simply hang around and wait and then reconnect. To my knowledge, there's no evidence that neurons actually regrow, so that there's cell division also at the neuron level. I don't know of any evidence of that.
I see. Thank you so much.
Okay, our next question is from Laurel Carney. Laurel, would you like to ask your question? Or
Yeah, sure. Thanks for the talk. It was great. Just sort of a curiosity question. do you think barn owls are special amongst owls? And I realize the same level of study hasn't been done on other owls, but the gross anatomy that you showed, revealed that huge basilar papilla. And I wonder if that's observed in other owls as well.
Ooh, there's not a lot of data. There's actually more on the brain. So the sound localization circuits have been looked at, or the midbrain, I should rather say, has been looked at. The famous space map that is there in the barn owl is not there to the same specialized extent in other owls. So it depends on the ear asymmetry. But that's the sound localization circuit. You're asking about the basilar papilla. Not very much data out there. I'm just trying to think. I should know, but I can't remember any remotely detailed studies on other owl basilar papillae. I don't even know the size. I would have to check, but I don't think there's even crude data.
Yeah, I thought there might be some skulls sitting around in museums.
There might be some CT scans in the meantime. I haven't checked in a while, so that might be an interesting thing to look at. There's a lot of publicly available CT scans nowadays, and the cochlea usually lights up like a Christmas tree in any of these because the bone is very dense. So it's very easy to see in a micro CT scan. I should check. Yeah, that's it. There could be some data out there in the meantime, but no real basilar papilla data that I'm aware of.
Thanks.
Okay, great. Are there any further questions? Okay, great. Let's thank our speaker one more time. And thank you, Christine, for a really interesting talk.
Thank you. It's been a pleasure. And thank you for all your questions.